Again, I am Alexa Tinsley. I'm the Director of Programs here at Jett Foundation. I want to thank you all for joining us today. We are welcoming Wave Life Sciences to discuss their interim results from their FORWARD-53 study of WVE-N531. We are so grateful to partners like Wave for helping us make all of these possible. And today we welcome Anne Marie, who is the Chief Development Officer for Wave here. So she will be presenting to you today. But before we get started, I do just want to mention a few things. These webinars are being recorded. So if you have to log off for any reason, or if you know someone that maybe wanted to attend this webinar and wasn't able to, it will be living on Facebook. We're streaming live, so it'll live on Facebook. It will also be recorded and uploaded to our YouTube for later watching, and you can share those as well. At the end of the webinar, we will be doing a Q&A. We do try to use the Q&A panel box. So there's a Q&A button at the bottom of your screen. It's a little chat box that has a question mark in it. So we do primarily use those for question and answer. If you do put it in the chat, we do try to get to it, and we also do try to get to our Facebook questions if there are any as well. But the Q&A panel, that little box that you type questions is our primary go-to for reading and answering questions. If for any reason you have a question that we may not get to, we will try to do our best to get them to Wave to get answers to you after the fact. We also know that people do watch these after as well, or it also takes time sometimes to process the information. So if you do have any questions after this webinar, please feel free to reach out to us at info@jettfoundation.org. So there's also closed captioning and language translation if that's something that you need. You can enable this by clicking the CC button and enabling transcription in your preferred language. If you are watching this after the fact, you can also do subtitles and auto translate. But that is all from me, and I will hand it off to Anne Marie.
Thank you so much, Alexa, and to the Jett Foundation for giving Wave and myself the opportunity to present to you today. I'm really happy to have the opportunity to share interim data from our FORWARD-53 program, and would like to just start by perhaps giving a little bit of history for Wave in DMD. So if you go to the next slide, we've been on a very long journey in DMD. More than 10 years ago, we began developing a molecule called suvodirsen. Unfortunately, our studies in suvodirsen didn't work. We had to stop that program because we found that suvodirsen was not producing any dystrophin. And we regrouped and went on to develop another molecule, N531, that I'm talking about today. So really delighted to be telling you more about the interim data. And we would like to thank the community for the continued support and really sticking with us through this journey. So when you have a failure in clinical development, the most important thing you can do is to reflect on what the data will teach you and how you can improve to make success more likely next time. So we go to the next slide. After the disappointing news with suvodirsen, we went back to the drawing board to assess how the animal models that we had used to predict whether suvodirsen would work, whether they were really giving us the best possible data to support translation of the molecule into the clinic. And we had used an animal model called the MDX mouse, and it's a naturally occurring mutation in mice. And these mice have milder symptoms than DMD patients do, due to the fact that the mice can upregulate a protein called utrophin. So instead, we decided to use a mouse model called the double knockout mouse, or DKO. And the DKO mouse has much more severe symptoms, and the mouse survives between 6 and 20 weeks. So it's more able to show us whether a potential investigational therapy might have the opportunity to change the disease course. So we tested a molecule that had the same chemistry as N531 in this double knockout model. And we saw a significant increase in both survival and an improvement in heart and lung function in the animals. And interestingly, as well as seeing more drug in the heart and the diaphragm, we saw more dystrophin in those tissues too, compared with the skeletal muscles of the mice. So this is very intriguing, because the aim of any DMD therapy ultimately is to prevent cardiac and respiratory failure. And this gave us a lot of confidence to move back into clinical studies. So we first completed a small clinical study in 3 patients to ensure the data were translating. And once we saw that, we expanded the study to 11 patients. And today I'm going to be reporting to you the results from those 11 patients. Next slide. So FORWARD-53 is an ongoing, potentially registrational, open-label phase 2 study of boys with DMD amenable to exon 53 skipping. And the key endpoints are safety and tolerability, pharmacokinetics, pharmacodynamics, including exon skipping, and dystrophin expression, measured after 24 and 48 weeks of treatment. Functional measures are being assessed at baseline and at 24 and 48 weeks of treatment. But since the disease progresses somewhat slowly in this population, 6 months of follow-up is inadequate to determine whether N531 is having an impact on slowing disease progression. The interim analysis was planned after 24 weeks and included pre-specified analysis to look at dystrophin protein in ambulatory patients. Next slide. So 11 boys were enrolled into the study. Their ages at enrollment ranged from 5 to 11 years. All the boys are on stable background steroid regimes, and there are 10 ambulatory and 1 non-ambulatory boy. And there were 5 different underlying mutations in the exon 45 to 52 region. Next slide. So I'll start by just talking about the safety and tolerability. So through 24 weeks of treatment, this looks very good. All the treatment-related adverse events were mild in intensity. There were no serious adverse events, no discontinuations, and no oligonucleotide class effects. It was, we think, an extraordinarily clean profile for an exon skipping therapy, which we do know is very important for patients and their families and prescribers, of course. Next slide. So turning to the results, we observed muscle tissue concentrations of 41,000 nanograms per gram, and this was consistent with our small experiment in 3 patients. And this also resulted in a tissue half-life estimated to be around 61 days and is supportive for future monthly dosing of this drug. So the boys in this study are actually moved on to monthly dosing now. And we observed a high level of exon skipping with a mean 57%, confirming that we have restored the reading frame for dystrophin. Next slide. So I'm showing now the dystrophin results from the patients across the study. 1 biopsy of the 11 boys was found to be too low quality to be analysed. So results from I'm showing here are from 10 boys. So firstly, and most importantly, we saw a highly consistent dystrophin expression across the FORWARD-53 boys, with dystrophin expression ranging from, you can see on the right here, 1, but with a mean of 14% of normal in mean muscle-adjusted measurement. We opened the study to both ambulatory and non-ambulatory boys. So the 1 non-ambulatory participant is excluded from the mean analysis, pre-specified analysis plan. And we reported all levels as MHC normalized. Actually, I misspoke earlier. It ranged from 14 to 1%, apologies. And the mean we reported is 9% in the muscle content adjusted measurement and 5.5% without muscle content adjustment. So our dystrophin was quantified from 2 isoforms consistent with those observed in Becker muscular dystrophy. Patients with this form of muscular dystrophy display a milder form of disease. I think really importantly, the dystrophin expression was also highly consistent, with 89% of the ambulant boys having levels exceeding 5% of normal, which is very encouraging. And this differentiates N531 from other approved exon skipping therapies. On the far right, you can see the boy who was non-ambulatory. That patient has about 1%, just over 1%. Obviously, this is lower than the ambulatory patients. However, we do also note that 1% is levels of dystrophin that are produced with some approved exon skippers on the market. And based on the non-clinical data, there's also a potential that more dystrophin is being produced in heart and lung. Next slide. Heart and diaphragm, sorry. So to achieve exon skipping, N531 needs to access the nuclei, and we confirmed that N531 is present in myofiber nuclei with red staining shown on the left of this slide. And once again, we detected N531 in myogenic stem cells in the majority of boys, as shown in the image on the right. And in this image, the PAX7 positive stem cells are shown in brown, and N531 is shown in red. So I'll just pause to remind everyone why it's important to see uptake of drug in myogenic stem cells. These are the progenitor cells that produce new myoblasts. And in DMD, the absence of dystrophin in muscle stem cells impairs their ability to divide and to create new cells. So this diminishes the ability of the muscle to renew and repair. And this means that it can't regenerate, the muscle can't regenerate. So restoring dystrophin in muscle stem cells is expected to enable their proper function. And this includes their ability to further undergo asymmetric division that is needed to preserve the renewal capacity of the muscle and to have productive repair of muscle fibers. So we're not aware of other clinical data for exon skippers or gene therapy that have been able to demonstrate myogenic stem cell uptake, and this is something that I think is promising for this therapy. Next slide. I'm going to now show you some slides from biopsies from the study. We compared biopsies from a boy who participated both in the short run-in study of 3 patients that I mentioned, and in that study, 3 boys received just 6 weeks of N531. So that's 3 doses over 6 weeks. That's shown on the left. And FORWARD-53 on the right after 24 weeks of treatment. And in both of these stained sections, we processed to show N531 localization, and we can see evidence of myocyte regeneration, as shown by the increased number of internalized nuclei marked in black arrows. So you can see from the left to the right, the number of black arrows have increased. And that really is a marker of improved muscle health. And these results are very encouraging to us because it's understood that when you're creating dystrophin, you should also be seeing changes in muscle of improved health and function. And you can see that the cells, the myocytes are more round and uniform from the left to the right. Next slide. So over the course of the study, we also observed clinically meaningful decreases in serum biomarkers that are also indicative of muscle health, such as CK and AST. CK significantly decreased from close to 12,000 units per litre to just under 6,000 units per litre. And similarly, on the right, you can see the AST decreased from almost 300 units a litre to just under 200 units a litre. So this reduction in CK is larger than what is typically seen when you introduce steroids to people who have DMD. But all of the boys in this study were already on stable background steroids. So this change is on top of steroids and much greater than what you would see in addition to the steroid introduction. The CK and AST results were highly correlated. And so it's encouraging, we think, to see these changes, these biomarker changes in muscle health emerging concomitant with seeing dystrophin starting to be expressed. Next slide. So in summary, we feel that this is really an encouraging data set at this interim time point. We saw highly consistent dystrophin expression across the patients with a mean of 9% muscle content adjusted dystrophin. This was quantified from 2 isoforms consistent with Becker muscular dystrophy and was from this weekly treatment. We also saw, moving to the right, upper right quadrant, really significant tissue concentrations and a half-life which is consistent with monthly dosing moving forwards, which we know will be important for people with DMD and their families. And the preclinical data would suggest that there's an opportunity for this benefit, a future benefit, to be to the heart and the diaphragm, but of course studies will need to be done to demonstrate that. N531 was safe and well tolerated in the study with no SAEs, no discontinuations, and no class effects. And we have seen at this interim time point evidence of both improved blood biomarkers for muscle health, localization of N531 in muscle stem cells, and improvement in muscle fiber regeneration, looking at the slides that we showed. So I really want to sincerely thank the DMD community for their support. And when we were designing the study, we took advice from the community, and we tried to incorporate the things that we heard into this design. And some of the things we incorporated included not running a placebo arm at this time, understanding, of course, that placebo is a very significant burden for patients, and latest confirmatory studies are required to have placebo, but at this stage, showing that the drug could produce dystrophin, it's not necessary, because unfortunately, people with this condition are not able to produce dystrophin. So in seeing that, it's already very significant. We looked to minimize the number of biopsies we undertook. So for the run-in part of this study of 3 patients, that's called part A, we only did 1 biopsy. And in FORWARD-53, we've done no baseline biopsies, and we're doing biopsies at week 24 and 48 to understand the amount of dystrophin that's being produced. We introduced trial concierge services to really try to support travel and support services. We know that it's very burdensome for people with DMD and their families participating in these studies. We introduced prepayment of reimbursement to try and avoid out-of-pocket expenses. And we introduced an option for home nursing where that was possible. And we are and remain fully committed to communicating our trial results, such as today, and working closely with global advocacy groups about the study and the results of the study, and providing ongoing communication and updates around the program. Now, one of the questions we frequently get is from people who are not currently eligible for N531 because of their mutation, and what our future plans are in regards to this. So we know that increasing dystrophin expression over approved therapies, could be a benefit for lots of other DMD forms. So we are looking at exon 51, 44, 52, and 45, with the potential to address up to 40% of the DMD population. And based on the interim data, we expect to engage with the regulatory authorities to discuss the next steps for this program. I would like to again sincerely thank all the boys, families, and clinicians and study staff who are currently participating in the study and enabled us to get to this interim time point. And with that, I would like to open for questions.
Excellent. We do have a few. Excuse me. So the first one is, can families with non-ambulatory children opt for a more frequent dosing, something like every 2 to 3 weeks instead of monthly dosing?
Yeah, that's a really great question. Thank you. So you saw from this presentation that we have very significant amounts of drug in tissue and in nuclei. So we think that there is adequate drug in the right place in order for the treatment to have its effect. In moving to monthly dosing, we're generating data that would aim to show that we're still achieving the right exposure of the drug in the tissue and the cells and the nuclei to continue to support dystrophin expression. The PK would indicate currently that that's possible, but obviously we have to demonstrate that with the study we're doing. And the lower levels of dystrophin in skeletal muscle for non-ambulatory patients is probably not so surprising, because obviously once muscle has atrophied and there's fibrosis, those myocytes are not able then to further repair and regenerate. But the non-clinical data would suggest that there is still capacity in things like heart and diaphragm. And so our assumption would not be that non-ambulatory patients need even higher levels of drug than we are already achieving. So if the data support monthly dosing, which it looks like it does, and the final results will confirm that, then the monthly regimen would be applicable to all patients who are eligible to take the drug.
Awesome. Thank you. How is the drug able to edit the muscle stem cells? How is Waves Oglio different from the other pharmas?
We think it's something to do with the uptake of the drug. So there's some special elements of the chemistry of N531, including its charge, which we think makes it more able to access the nuclei. And we think this could be why N531 is accessing stem cells. Of course, we can't know for sure, but what we can know is we can see it in the stem cells, and that hasn't been shown with other therapies.
Awesome. What about other exons in the pipeline? Are there any plans to focus on double skipping, for example, skipping exon 6 and 7?
I think this is a great question. So when I talked about the other exons, obviously there are still, many other people with DMD who are not unfortunately not covered. And one of the things that is very encouraging from the FDA is they have put out guidance around a concept of doing what's called a platform study. This means that you enroll people with lots of different mutations into a single study, and you share a placebo arm, and the placebo arm is also like mixed of many mutations. So this would give the opportunity to study other rarer mutations at the same time. So I can't speak specifically to those, but I think the way the field is moving, it's going to become much easier for this research to be done to support people who have rarer mutations in receiving therapies in future.
Awesome. Looking at the preliminary data, would it suggest that if taking N531, the requirements for steroids would be removed?
Yeah, that's a great question. So in the FORWARD-53 study, all of the patients are on background steroids. So the results that we are showing, and hopefully will show through the final readout, will be in addition to steroids. However, with higher levels of dystrophin and potentially in future showing, we would hope that this results in slowing of the loss of function for patients. It does create an intriguing possibility in future as to whether steroids could be tapered or removed. And that would be a study we would need to do to demonstrate that. But, presently, everybody remains on their background steroid treatment in the absence of evidence that shows they can do without that.
Awesome. And then based on that feedback, are you going to be looking at steroids as a whole, or are there plans to open a separate cohort with vamorolone or Agamree?
So at the moment, we are taking patients who are on any steroid, including vamorolone and others, and we just require that people have a stable background regimen so that we can really see what the difference is of N531 treatment on top of that. So if we do a future study to look at steroid withdrawal, we probably would approach that in the same way rather than teasing out individual steroids. But to your point, there are now next generation steroids that are more effective. So that's to do something like that, you would probably want to look at, some sensitivity analyses to really find out, is there any difference between one steroid versus another when you withdraw?
Thank you. And then I'm not sure if you have this answer, but when is phase 3 of the trial likely to start?
Yeah, that's a really good question. So FORWARD-53 is a potentially registrational study. And as I mentioned, we're consulting with regulators and the study is due to finish in Q1 of next year. So the next phase for this program would actually be filing for an NDA and actually conducting a confirmatory study. That's required under what's called the accelerated approval pathway. So this is a registrational pathway that FDA has in place that allows sponsors to make an application for a license to market a drug using a biomarker like dystrophin. And one of the conditions of that pathway is that sponsors will conduct a confirmatory study, but this is a post-approval commitment. So that study would be initiated, but would not need to be completed for N531 to be submitted as a new drug application.
Thank you. And then last question that I have is on one of the slides, it states exon 52. 45 to 52, and other exons have been studied. Would one missing 46 to 52 be considered for this therapy at this time?
The study is not recruiting currently, and ultimately, the likely patient population that will be indicated will be encompassed by what we have studied. But that will remain to be seen what FDA will eventually decide, which patients they would support us treating.
Perfect. I'll give it another minute. If anybody has any questions, please feel free to put them in that Q&A box. I don't see any on Facebook at this time, but again, if you do have any questions after you've processed the information, or if you're watching this after the fact, please do feel free to reach out to us at info@jettfoundation.org. I want to thank Anne Marie and the entire WAVE team, and also all of you for being here with us this morning to learn more about N531. So again, we appreciate all of you. We hope to see you again very soon. And again, if you have any questions, please feel free to reach out. Have a great day, everybody.
Thank you. Thanks so much. Bye.
Bye. Thank you.